Thank you everyone for joining. So today I'm going to be talking about day one of the Tapestri workflow. You will notice here it is Tapestri DNA sequencing workflow. So some of you know we also have a multi-omics workflow, which is DNA plus protein, but I am not talking about DNA plus protein today. There is a talk scheduled the 4th of June for that. So we will go over the specifics of DNA plus protein. Today it is only DNA. If you're interested in that talk, we can share that link with you, of course, after. So let's just go through the agenda quickly. So as Kelly said today, we're just going through the steps of the Tapestri workflow. So these are encapsulation, lysis, barcoding, targeting PCR, and then we will take a little bit of time to talk about multi-sample workflows. This is just a screenshot of the Tapestri DNA sequencing protocol timeline. So I'm not sure if you had the chance to join the session that one of my colleagues did last week. That was all about cell preparation. So with the Tapestri platform, you can get from cells in suspension or isolated nuclei all the way to library readies, sequencing ready libraries in 1.5 days. And the total hands-on time is around 5.5 hours. So my colleague talked about sample preparation, how you get your cells in suspension, or how do you isolate your nuclei. So that's why I am not touching on that today. I am starting from the encapsulation part all the way to the targeted PCR, which usually runs overnight. And then after the AACR session, we will have another seminar, the support team, to talk about the second day of the Tapestri workflow. So as you can see, the hands-on time of day one is very short, and we will see the steps step by step. Let's just see quickly which steps are done on the Tapestri platform and which ones are done on the bench. So as I said, cell prep, I'm not talking about that today. That is, of course, done on the bench. Then you have encapsulation and cell barcoding, which are steps done on the Tapestri platform. And then lysis and targeted PCR of the Tapestri platform on a thermocycler. That's for day one. And then you have day two for library preparation and day three for sequencing, although you could even start the sequencing on day two at night. Before I start, just telling you we have 2 different protocols available for this assay. All of these protocols are available on our website. I'll give you the link at the end. We have a long protocol, a long user guide with all the details you require to know about the Tapestri platform, how you prepare your cells in suspension, troubleshooting guides, pictures. It is very comprehensive and longer. And we also have a quick reference card or short protocol that it is very handy for people who have already run the assay, or people who don't want to carry such a big user guide to the lab with all the essential details anyway to run the assay for the Tapestri platform. Both of them are all available, on the website. Let's just then focus on the workflow, and I'll give you a review of the whole process. So the sample input for the Tapestri platform is 3,000 to 4,000 cells per microliter in 35 microliter, which is equivalent to around 100,000 cells. So you start with 100,000 cells or nuclei, and then you use the Tapestri platform to do the encapsulation and barcoding. This is all we will talk about today, the assay. But then so you know that this workflow fits very nicely in NGS pipelines, existing NGS pipelines, because once you get your sequencing ready libraries, you sequence them. We are compatible with all Illumina sequencers, all Illumina platforms. And once you get the data, you could use our software to analyze it. So we will have sessions on that. Let's just focus on what happens inside the Tapestri platform. So we work with disposable microfluidics cartridges, and this is what it is depicted here in the cartoon below. So the next step, the first step is the encapsulation. Here we are encapsulating single cells or single nuclei with a protease solution in 40 micron oil droplets. This process only takes 5 minutes, and we follow a 5% Poisson distribution. This means that we will generate millions of droplets. Majority of them will be empty, and only 5% will contain a cell. This minimizes the chances of 2 cells being encapsulated together, and that's why we do the 5% distribution. At the end of this step, we will have a nice emulsion with our single cells, right? And each of them will be inside of a droplet. But we have to digest these cells because we have to get rid of all the proteins that sequester our genomic DNA and hinder the molecular biology downstream. So that's the lysis step, and that's done outside on a thermocycler. So we digest all these proteins and we have our genomic DNA freely of a single cell. And it is after this digestion when we again use the same, but different parts of the same cartridge to combine this cell lysate with barcoding beads and all the necessary reagents and primers to perform PCR amplification overnight. So essentially, at the end of this process, which is called barcoding, and this takes 45 minutes, you will have picoliter PCR reactors where you will have the genomic DNA of a single cell, a barcoding bit, so a unique barcode, and all the set of primers of your panel of interest. So if you are working with acute myeloid leukemia, you will have your panel, all the amplicons, all the reagents will be there on that picoliter PCR reactor, and then overnight, you will be amplifying all of that at a single cell level. I don't know if there is any question at this point. You can ask me at any time. But yeah, essentially, this is what happens during the barcode. And because the barcoding bits has a barcoding step, and the barcoding bits have a unique barcode, overnight you will be amplifying that. And the day after, which is the second day of the assay, each DNA strand that you amplify from a single cell, it's already barcoded. So that's why you do general library PCR the second day. Right, so let's just move on to encapsulation and see what happens in the lab. So as I said, we work with disposable microfluidics cartridges, and these cartridges is where the magic happens. So when we are in the lab, the FAS team are very careful and emphasize so much in the fact that we have to be very clean manipulating these cartridges. That means, you know, being out very far away from like a ventilation system, or avoid dust or debris to clog any of these channels. We manipulate them by the walls, and we use one cartridge per sample. So this is a picture of a real cartridge, and what you see below is the base plate. So the cartridge sits on top of the base plate. And you see that tube over there? That tube is where we will collect all the emulsions that we generate during the encapsulation step, which takes these 5 minutes. So as I said, we're always very careful to not contaminate the cartridge, and we specify people to work with the cartridge on the base plate inside of the Tapestri instrument to pipette the reagents. And this is what you see here in this picture. So you have the cartridge on the base plate inside the Tapestri instrument, and in this way, when you pipette the reagents, you avoid any sort of dust or debris that could come from above. The manual is very straightforward. It is all color coded. You see on the right the reagents you need for the encapsulation step. So you need the lysis buffer with the protease. You need your cell suspension. In this case, it's 3,000 to 4,000 cells per microliter, and encapsulation oil. And this is what happens. So here we are combining again the cells with the protease, and with the encapsulation oil, you see the droplets being generated. So at the end of this process, which takes 5 minutes, what you would expect is this specific tube. You will have oil at the bottom and then a white layer of emulsion on top. This is what you will expect after the encapsulation step. So once we have pipetted all the reagents here onto our cartridge, we just have to use the Tapestri instrument to run the program. So it is a very easy to use and intuitive instrument, the Tapestri platform. It has only 3 programs available: encapsulation, priming, and barcoding. And priming and barcoding are part of the same program. We will see now in a couple of slides. For encapsulation, you just have to select the step, and then it runs a 5 seconds quick test, and then a 5 minutes program. So at the end of the encapsulation step, this is what you should see. This is the tube that I mentioned before. So you should see a tube with the oil at the bottom and then a white thick layer of emulsion on top. This is what a high quality emulsion looks like. And this is what we usually expect. Sometimes we see no emulsions, something might have gone wrong. One of the reasons why we could see no emulsions generated after the encapsulation step is perhaps some contamination with debris or dust, that if it clogs one of the channels, then all the reagents will not flow, or some of the reagents will not flow, and we will not generate successful emulsions. But what we see normally is that we generate high quality emulsions on the left. So this is the second. Yeah.
We have a question. These are really cool droplets. How many droplets are made during this process?
Millions. I wouldn't be able to tell you from the top of my head, but 2 million, I would say, ballpark.
Great. And then you mentioned it's 3,000 to 4,000 cells per microliter. What is the doublet rate at that concentration?
Yeah, that's a good question. So the doublet rate at that concentration is around 5 to 8% doublet rate. And actually, so the doublet rate is not so much because of this encapsulation step, because we take good care, good care of doing the 5% Poisson distribution. It is perhaps more due to handling, and that is what the next step I'm going to talk now. But yeah, the percentage of droplets is between 5 to 8%.
Great, thank you.
And we will see that. Yeah, no problem. So we see that if you work with known cell lines, for instance, you could see and calculate the estimated percentage of droplets. So this brings me to this point, actually, a very good question, a very good time, because we take good care of the encapsulation to avoid cells being encapsulated together. And we also have to be careful when we handle the emulsions. So this is the second step, when the FAS on site emphasize so much our users to be careful with the emulsions. We have to handle the tubes by the lead, and we have to place them carefully on a rack when we work with them, and absolutely avoid touching the sidewalls. Why? Because these emulsions are very sensitive to surface tension. So if we touch the surface walls with our fingers, those droplets could merge. And then if the droplet merge, then that could potentially be a source of creating more doublets, right, that we want. So that's why we have to be very careful in manipulating that. And that's why the FAS go on site, and we perform a 2-day training to make sure that all our users know how to handle the emulsions. Right, so after we have finalized our encapsulation step, we are ready for lysis. But before, there is one step that we have to perform in order to have a successful lysis, and that is removing a little bit of oil from the tube. So let me just play this video on the right at the same time. So the way we remove the oil is by using a gel loading tip, and that's because they are very thin, right? So we have to go through the emulsion to remove the oil from below. The thinner the tip, the better. That's why we use gel tips. And that's the pipetting technique, what we are showing here. So why do we remove the excess of oil? Because we want all our droplets to be within the 100 microliter limit of the thermocycler. And this volume that you generate is generally higher. So we just remove a little bit of oil, and what you would expect after oil removal is something like this, the picture on the right. So again, all of this, we do it during the training on site. So now after you have removed the oil, you are ready for lysis. So you just place your tube on a thermocycler, and to ensure even temperature distribution, we ask our users to use either a skirt, or if they don't have a skirt, just place tubes in the corner to ensure even heat transfer. This is the program that we run, very simple, 50 degrees, 60 minutes to digest, and 80 degrees, 10 minutes to inactivate that protease. And you can see here the picture of exactly all the proteins that we are getting rid of. So if people wonder why the protein treatment is important and the protease treatment is important, I always love to show this graph over here. So what you see on the left is the percentage of barcodes, hence percentage of cells. And on the x-axis, the cells with over 20x read coverage per amplicon. So the blue bars are with protease treatment, and the red bars are without protein treatment. So that's why we see protease, sorry, not protein. So that's why we see that if we didn't do this protease treatment, we will not generate data at all. So this step is very critical and very important to ensure efficient PCR amplification. Right, so after the protease treatment, we are ready for barcoding. But before barcoding, notice that we haven't done much hands-on time so far. So before barcoding, we have to prepare the cartridge. So this is the same cartridge that we use for encapsulation, but just different reservoirs we are using now. We have to prepare the cartridge in order to have it ready for the combination of the droplets, the oil droplets that we have just digested, with our barcoding primers and reagents. So for that, we use electrode solution that we just pipette on those wells over there, and that run the program, and that's 20 minutes hands off. Ideally, we want this program to be run immediately before the barcoding program. And then that's it. So after we have finished the priming, we just go through barcoding. So again, notice the color code on the right. You see all the reagents you require and then which reservoirs. Again, it is a different part of the same cartridge, right? Cell Lysis, which is number 6, is your tube, the tube that it is on the thermocycler, that now those emulsions, you pipette them back to reservoir number 6. So again, just a reminder, what is barcoding? Barcoding is a combination of your cell lysate from your thermocycler with barcoding oil, barcoded beads. These beads are a carrier that bring that unique barcode, and the barcoding master mix. So all the primers for your panel or the enzymes and the reagents that you require. And what you have here are these picoliter reactors. So all the ingredients you need to perform multiplex PCR overnight. So you might notice that instead of having one tube, now we have 8 tubes. The reason why we have 8 tubes is because the volume that we generate during the barcoding step is larger than what can fit on a 0.2 PCR tube. So we just divide the solution across different 8 tubes, but these are complete replicates. What we see sometimes is the volume of these tubes is uneven. That is normal, completely normal. You could see less or more volume of oil or emulsion. That is due to cartridge architecture. But even if you saw one tube which is empty, that would sometimes see, it doesn't matter because the total amount of emulsion generated has been divided across the remaining 7 tubes. So that's generally no problem at all. This process takes 45 minutes, and it's 45 minutes hands off until you have these 8 tubes with your emulsions.
Emma,
and again,
question.
Yes.
So while you're waiting for the first incubation to activate the protease, how would you suggest storing this very precious cartridge until you get to this step?
Yeah, very good question. So actually, when we finish one step, you could either remove the tube while you are removing the oil and put it on the thermocycler. You could leave it inside of the Tapestri instrument and close the lid, so there is no risk of contamination with debris. Or some people also take it and use the same bag and just, keep it protected with the same plastic bag just to avoid any sort of contamination. Either you can do. I generally leave it on the Tapestri instrument. So you manipulate it less, you don't touch that cartridge unless you have to.
Fantastic.
Perfect, good. So after we have finished this barcoding, we have our little PCR reactors. This is super precious. At this point, you don't want to break these emulsions. You never want to break them. But of course, now you have everything you need for a successful amplification. But before you move to PCR, of course, you have to also remove the oil. So I'm not putting the video here because we have already seen it. It's exactly the same process, but now repeated 8 times. Why? Again, just because the volume of oil and emulsion is larger than 100 microliters. So there is a special note here that I like to add. So I know people usually ask, as you have H tubes, this is actually a real sample of a successful barcoding. You see there are perhaps uneven volumes, that's no problem at all. Some people ask if they could use a multi-channel. We rather ask them to use a single channel because you work with these gel loading tips, and we feel like it is better if you are more careful doing it single channel. Again, what you are expecting is to have all your H tubes looking like the picture on the right after oil removal and all the emulsions within the 100 microliter. And now is when you are ready to put your tubes on a thermocycler and do targeted PCR. So again, same rules apply. On the left, you should use an skirt or tubes in the corners to ensure even distribution. Make sure they close well. These programs are long. You can see on the right, the time varies depending on the number of amplicons, but minimum is 2 hours. It could go up to 4 or 5 hours. So we really want to make sure that the heat is even and there is no evaporation. So usually, just to give you a ballpark of time, if you started your experiment at 10:30 or 11, by 3 p.m., you have your PCR overnight, and you have done 20-25 max hands-on time for day one. So this is when your day one will finish, and you will continue on day two breaking the emulsions, because these emulsions in day two are not so important anymore. Each DNA is being labeled with the barcode. So if you break the emulsions, it doesn't matter because you will know which cell this DNA is coming from. But I leave you that for the next seminar. So at this point is when you will finish day one. And then I just want to take a little bit of time to discuss multi-sample workflow in case you are wondering whether you could do more than one sample a day. So that's the case. We could do with one instrument and one operator up to 12 samples per week. And that would mean you do 3 samples a day, Monday to Thursday, and you do the library of all of them on Friday. So just a quick reminder of the assay. So you will do, if you wanted to do 3 samples at the same time a day, you will do the cell prep for all of them together, and then you could encapsulate. Blue steps are done on the instrument and then red on the thermocycler. So you could encapsulate them back to back. This is only 5 minutes, so the emulsions can wait. You encapsulate one sample, then the other sample, and the other sample, and you put the 3 together to lyse to digest on the thermocycler. And then you take one by one and you do priming, barcoding, PCR. And this waits on the thermocycler at 4 degrees. When this is done, priming, barcoding, and PCR, and so on. So it is ideal that you have 3 PCR machines to do that, because as you see, the targeted PCR overlaps at this point. But, and even though you are doing 3 samples a day, even though if you're doing 3 samples a day, it all follows into a working day from 9 to 5. So you could actually do many more samples if you wanted to. And then on Friday, you can just combine the library prep of all the samples together. And it is not very cumbersome, because even if you get 8 tubes from the barcoding, after PCR you pool. So you don't have many tubes, even if you are working with many samples at the same time. So that is all I had from my side in terms of day one and multi-sample workflow. I just wanted, before I finish, to remind you of our Mission Bio support knowledge base. This is a great website and resource where you can find information about everything: sample, Tapestri, instrument, experimental workflow. So all the questions and details we have gone through today are there. Designer, how to create your panel. So have a look if you have questions about the Tapestri platform. And if you don't find what you're looking for, of course, you can also contact the local FAS. So with that, I have finished. Thank you for your attention. Let's see if we have any more questions.
Emma, thank you so much for the in-depth look at all of these processes. It was incredibly enlightening. So we have a few questions about the PCR thermal cycling. So this attendee noticed that the ramp rate changes, and they're just wondering why and why that's important.
Yeah, very good question. So so when people ask us what thermocycler they should use, we don't generally give a brand as long as it can go 1 degree. So from 4 to 1 degree ramp rate. And that's very important. And the reason why we have the 1 degree ramp rate is because it is better for emulsion stability. Our R&D teams have checked different protocols, and they have come to the conclusion that 1 degree per second ramp rate is better for our emulsions, and we know that we have to take good care of them. So that's why we use 1 degree ramp rate. So it is important, and we ensure when people are going to purchase a Tapestri platform and do the training, we ensure beforehand that their thermocycler could go down to 1 degree.
Great. And then a similar question, can you explain just a little more why you need different times and how do you decide what time to do for your panel?
Yeah, so for users, I mean, we have highlighted the AML here because that is the assay we use for training. That's why we have it highlighted. How to decide, it will depend on the number of applicants. So here on top, you see we have different annealing times for panels 20 to 100, 100 to 200, and so on. The reason we have different annealing times is because we want to allow enough time the primers to find their preferred sequence, let's say. So that's why the larger the panel, the longer the time.
Great, fascinating. And then one final question, if you could give one piece of advice for this part of the protocol, what would it be?
For this part of the protocol specifically, ensure beforehand that it is true. I mean, what we just discussed. So the ramp rate is really important. We have to make sure beforehand that can be met to ensure success of the assay. And I couldn't think, apart from the skirt and the corner tubes, I couldn't think of anything else apart from ensuring that the time is, the ramp rate and the program is correct.
Okay.
For this specific step, if you ask me for others.
If there will be variation when using different thermal cycling instruments, testing multiple samples on the same day, will there be variation if you use different thermal cyclers?
We would need to look at that. So we have to make sure, because we have found that different thermocycler brands have different ways to set up the ramp rate. And even, so we have to check if people are want to do a multi-sample workflow and they need to run 3 thermocyclers at the same time. Ideally, we want the thermocyclers to be the same, but that's not obviously possible sometimes. So they have different brands. If they have different brands, we will spend a little bit of time in talking to the vendors and making sure that the programs really tally. So the programs have to take the same time, you know, and they have to have the same ramp rate. And I say ramp rate because some thermocyclers are super easy to program, and the ramp rate can be just set by degrees, but other thermocyclers work with percentages. So that's why we have to talk to the vendors or the support team of which of these companies to make sure that we are giving exactly the same ramp rate and total time, because you don't want any variability because of the thermocycler.
All right. Well, it looks like those were all of the questions. Gema, I want to give you a big thank you for doing our office hours this week. And for those of you who have attended, thank you again. And if you have any questions, please reach out to us at support@missionbio.com. And again, we'd like to highlight, we hope to see you next week at the AACR virtual conference. Stay safe where you are, and Gema, thank you again.
Thank you so much for joining. Thank you, everyone.